أعزائي الطلبة، اليوم أكمل معكم المحاضرة الثالثة من موضوع الهرمونات الإندوكراين، وأعتذر لوجود خلل في النت فاضطررت لأكمل المطلوب منا خلال هذا الأسبوع، وهو بواقع 3 محاضرات لكل أسبوع. فالآن نبدأ بالمحاضرة الثالثة هي GIT Hormones. GIT hormones هرمونات الجهاز الهضمي is regarded as the largest endocrine organ in the body. Collectively, GIT hormones influence motility الحركة, secretion طرح أو الإفرازات, digestion الهضم, and absorption in the gut, وكذلك الامتصاص بالقنوات الهضمية. In addition, they regulate bile flow, secretion of pancreatic hormones, affect tonicity of the vascular wall. tonicity معناه الشدة, المناعة, المقاومة, الليونة في الوعاء الدموي, blood pressure and cardiac output. The principal GIT hormones include وتشملنا على Gastrin, Secretin, cholecystokinin, pancreazymin, vasoactive intestinal polypeptide VIP, and gastric inhibitory polypeptide, and glucagon-like peptide, and the first hormone, الهرمون الأول الذي سنتكلم عنه, is called gastrin. There are 3 molecular forms of gastrin that are present in the blood circulation and the tissue, and these are هناك 3 أنواع من الجاسترين وهي موجودة في الدورة الدموية وتشملنا إلى ما يلي: big gastrin الكبير, which is a linear polypeptide of 34 amino acid called G اختصارا من الجاسترين 34 الذي هو أمينو أسيد. Little gastrin, which is a linear polypeptide of 17 amino acid, which is called G17, and mini gastrin, which is a linear polypeptide of 14 amino acid called G14. However, they all exert the same biological effects on the target cell. كلهم يعملون نفس العمل على الخلية المستهدفة. Gastrin is secreted and stored by endocrine cell called as G cell, mainly in the antral mucosa of the stomach, and to lesser extent by G cell of the proximal part of the duodenum, and by D cell of the pancreas. Following secretion to the bloodstream, the main circulating form is G34. Gastrin has the following main functions. وظائف الجاسترين الأساسية سأعرضها الآن بالسلايد اللاحق. Stimulate the fundal parietal cell to secrete the gastric acid. Stimulate gastric mucosal cell to secrete pepsinogen and intrinsic factor. Stimulate small intestinal mucosal cell to secrete a hormone called secretin. Stimulate the secretion of the pancreatic enzyme and bicarbonate. Stimulate gastric and intestinal motility. Stimulate gastric mucosal growth. Stimulate hepatic bile flow. The main stimulus for the release of gastrin is antral dilatation. Due to the presence of food in the stomach, mainly protein, digestion product, polypeptide, peptides, and amino acid. In addition, other stimuli may include هناك بالإضافة إلى ما ذكرنا من توسع القنوات في منطقة الأنترم بالاستومك، الحقيقة هناك عوامل أخرى تساعد على الإفراز، وهي الكحول، كافيين، insulin induced hypoglycemia, and vagal stimulation, which may occur by sight, smell, taste, thinking. All these factors create vagal stimulation, which lead later on to stimulate gastric secretion. Maximum secretion of gastrin occur at antral pH of 5 to 7. However, when the pH gets lower, a negative feedback mechanism will inhibit further secretion of gastrin. The second hormone is cholecystokinin pancreasamine. It is 33 amino acid polypeptide hormone released from upper small intestinal mucosal cell, which is called the eye cell, mainly in the duodenum, in the response to first, presence of partially digested protein products in the lumen of the intestine, entry of gastric acid to intestine, and presence of fatty acid, mainly in form of microcell. As the name implies, كما في معنى الاسم, it stimulates the gallbladder contraction. So it is called cholecystokinin. Chole is the gallbladder, and meanwhile the secretion of pancreatic secretion. So it is pancreasamine. That is mean stimulate the secretion of the enzyme from the pancreatic cell. It is present in multiple molecular forms. The most active forms is that containing the sulfated tyrosyl residue. The 5 amino acid sequence at the C-terminal end are identical to those of gastrin. The structural similarity with the gastrin make cholecystokinin pancreasamine exert some gastric action, mainly in the stimulating intestinal motility and increase pancreatic enzyme. It competes, that is من يتنافس, with gastrin for its intestinal receptors, and this may help in terminating gastric action following mean. بمجرد ما نكمل الأكل ويبدأ يشتغل سينتهي عمل الجاسترين. The other enzyme is secretin. It is polypeptide with the structural similarity to the VIP, GIP and Glucagon الذي لاحقا سنتكلم عنه. It is released from the granular mucosal cell S, that is mean secretin, S cells that are merely present in the duodenum, but however are distributed throughout the small intestine. It is called as such because it permits the good flow of pancreatic juice following meal. Secretin release is stimulated by contact of S cell with the gastric acid. However, it is only inhibitor is somatostatin. The principal function of secretin is increasing pancreatic juice release with the increased concentration of bicarbonate. دائما نتذكر بأن أكثر محتويات البنكرياتيك جوز هي البايكربونات، ومن اسمه secretin يساعد على السكريشن. So it is used for testing pancreatic function. لهذا هو يعطينا فكرة عن عمل البنكرياس. It inhibits gastric release and thus gastric acid secretion. The other hormone is the vasoactive intestinal polypeptide, VIP. It is a linear polypeptide of 28 amino acid, has some structural similarity with secretin, GIP and Glucagon. It is present in almost all body tissue, but mainly concentrated in the GIP and the nervous system. It is present in the nerve fiber along all the gut from the oesophagus, gut هو GIP, to the oesophagus, from oesophagus to the colon, but mainly in the jejunum, ileum, and the colon. GIP has a large number of ill-defined, non-specific physiological effects, among which are هناك له وظائف مختلفة وأهمها ما سأذكره الآن. First, act as a neurotransmitter in the CNS and in the autonomic nervous system of the gut. 2. Relaxation of the smooth muscle fiber in the vessel wall, gut, and genitourinary system. 3. Increase pancreatic secretion. 4. Increase water and electrolytes secretion from the gut. 5. Inhibition of gastric secretion and thus gastric acid secretion. 6. Increase hormone release from the gut, pancreas, and hypothalamus. 7. Stimulation of lipolysis, glycogenolysis, and bile flow. Gastric inhibitory polypeptide. It consisted of 42 amino acids released from the K cell, which are located in the jejunal and duodenal mucosa. Its main function include أهم الوظائف له تشمل ما يلي. First, in the presence of hyperglycemia, like following meals, it stimulates the secretion of insulin, This is the most important action of GIP, or the gastric inhibitory polypeptide, and that is why it is called the glucose-dependent insulinotropic peptide. فإذا هو له علاقة بعمل الجلوكوز وكذلك بالإنسولين. Second, decrease intestinal motility despite increased intestinal secretion of water and electrolyte. يعني من ناحية يبطئ الحركة، ومن ناحية أخرى يزيد من طرح الوتر and electrolyte into the gut. Third, in the supraphysiological concentration. من نقول في supra that is mean أعلى من عمله الفسيولوجي. It acts to inhibit gastric acid, pepsin, and gastric secretion. Glucagon-like peptide, it is a linear peptide, polypeptide chain that is released from the small intestinal mucosal cell, which is called the L cell, mainly in response to elevated blood glucose level soon after meal. It has some structural similarity to the glucagon, however their action are totally different. ونحن مثل ما نعرف الجلوكاجون هو عامل أو واحد من الهرمونات الذي يزيد إفراز أو يزيد الجلوكوز، فإذا هذا أيضا يعمل بنفس العمل لكنه بميكانيكية مختلفة. It acts to lower blood glucose level by mechanism similar to those of GIP, that is reduce GIP motility and gastric acid pepsin secretion, in addition to affecting the satiety center. satiety center هو مركز الشبع in the brain to decrease food intake. يخلينا نشبع حتى لا ناكل مزيد من الأكل. However, its main action is stimulating pancreatic beta cell to release further insulin in response to high blood glucose. من يزيد عندنا الجلوكوز يزيد عندنا الانسولين، وأيضا يزيد عندنا هذا الغاية منه to decrease هالجلوكوز الذي أصبح عاليا بعد أخذ الوجبة الغذائية to an extent that may even result in beta cell mass hypertrophy. ومثل ما تعرفون البيتا سيل هي الخلية الموجودة داخل الآيلد أوف لانجرهام بالبنكرياس، وهي المسؤولة عن طرح الإنسولين والمسؤولة عن تخفيض نسبة الجلوكوز في الدم. GIP and GLP-1 role in the treatment of diabetes. Both incretin، حقيقة يسموهم incretin، هؤلاء الذي اسمهم GIP and GLP-1 are of optimum benefit to control blood sugar. However, their major problem is that their half-life in the circulation is very short, and they are soon degraded by enzyme طالما هي يعملون على تنظيم نسبة الجلوكوز، لكن مشكلتهم أعمارهم أو نصف أعمارهم صغير جدا، لأنه هناك إنزيم اسمه dipeptidyl peptidase 4 or DPP هذا يعمل على تكسيرهم which are present on almost every body tissue. وهذا الإنزيم الحقيقة كلش مهم، واليوم العديد من الأدوية الموجودة في السوق وهي الأدوية المتقدمة جدا في علاج السكر هي تعمل على هذا الإنزيم الذي اسمه DPP4 or dipeptidyl peptidase 4. فدي P الأولى والبي الثانية 4. For this reason, any inhibitor to this enzyme, which is called the DPP4 inhibitors, have been synthesized as a potent drugs for type 2 diabetes mellitus T2DM, like sitagliptin or Januva بالاسم التجاري 100 ملغ tablet to prolong the half-life of incretin, enable them to stimulate insulin secretion efficiently. This drug has been used as additive therapy to control type 2 diabetes mellitus. Successfully. Moreover, synthetic analog of GLP-1 have been used in injection form for the control of hyperglycemia in type 2 diabetes mellitus. Example, exenatide and other. معنا إلى أسماء أو هذه المشتقات والأدوية الآن موجودة في سوقنا التجاري. سأعرج أيضا على بعض الهرمونات الموجودة في GIT. وother hormone of the gut include somatostatin. It is one of the most potent, well-known inhibitory hormone for a lot of endocrine secretion. It inhibits the release as well as the action on the target cell of a lot of hormones including Gastrin, Secretin, GIP, insulin, Glucagon, very important peptide VIP, Pancreatic Polypeptide, and other. The other hormone is the motility. It acts to release the contraction, increase the contraction of smooth muscle. من اسمه motility, that is mean something related to the motility, إلى الحركة. In the upper GIT, namely the lower esophageal sphincter, gastric fundus, antral and duodenum, it is unique feature. بنقول unique feature هي خاصية منفردة له, is that its action are restricted to fasting state only. فهو يعمل فقط عندما نكون في حالة the pancreatic polypeptide PP. It is mainly secreted from the pancreatic F cell. It acts to increase and then decrease biphasic action, has both increase and decrease, so it is biphasic action, the pancreatic secretion of water, electrolyte and enzyme. Its high blood level can be used and as a landmark of pancreatic endocrine tumor. Glucagon, it's a group of substances that are released from the gut but have biological effects similar to pancreatic glucagon. That is catabolism or catabolic to hyperglycemia, although are chemically different.